of the subkingdom Cryptogamia. Cryptogamia of the kingdom Plantae. Which split the Thallophyta into subdivision Mixothallophyta. Mixothallophyta, Euthallophyta. These are the main important kingdom Plantae, subkingdom Cryptogamiae, and next one division is a Thallophyta, subdivision Mixophyta, subdivision 1 Mixophyta, subdivision 2 is called Euthallophyta. Okay. Uh, once once bacteria Once bacteria, lichens, and myxomycetes were included among the fungi, though bacteria were removed from fungi. Okay, the myxomycetes continued their inclusion among fungi for reason other than biological. These are the studied by the mycologist by tradition and preserved the traditional and tradition and preserved along with the collection of the fungi. One second, I'll explain. Sorry for the interruption. Uh, the classification of fungi here, you can observe the both in based on the classification. Thallophyta is the most important, but the different organisms, these are the studied by mycologist. Okay, these are the studied by the mycologist, tradition and preserved along the with collection of the fungi. Tradition and with collection of the fungi. Main important, uh, one second. Sorry for the interruption. Now we will continue. Here, main based on the taxonomy, here Alexopoulos and Minis proposed fungal fungal classification in 1979. Okay, they places the fungi include the slime mold in the kingdom Mycete, the super kingdom Eukaryote with the addition includes the other kingdoms. They divided the kingdom Mycetes into three divisions. One is a gymnomycota, mastigomycota, amastigomycota. The subdivisions are these are the subdivisions. But this one, first one is the division one. Here, the true fungi divided Sacrodo in 1866. Four classes is there. One is the Phycota, Ascomycota, Basidiomycotina, and form from class Deuteromycetes. And the basis of the presence of or absence are septa and character sexual productions of the spores. This division, gymnomycota, includes subdivision here in that in this one, two subdivisions are there. One is the gymonomycotina, second one plasmodio gymnomycota. Acrisio-gymnomycota includes the single only one class. You can observe that one Acri-isomycetes. In this one, one class is there, Acromycetes. Here, the fungi of spore flagellated cell except one species. The class compresses. In this one, subclass 1, Acromycetade. Second subclass, Dictyosteliomycetidae. This is the subdivision, Acro-isogamomycotina. The second subdivision, Plasmodiogymnomycota. It is divided in two classes. This divisions are how many classes? To divided two classes. One is the Protosteliomycetes, the class 2 Mycomycetes. Here, Mycomycetes includes the true slime mold and comprises three subclasses. That is the first subclasses, Ceriatio, Mycomycetidae. In this one, only one order is there. That is the Ceratio myxetales. Subclass 2, Mycogastriomycetidae. Here, four classes you can observe. One is the lyciales, econisteliales, trichoales, physarales. These four subclasses you can observe to the Mycogastriomycetidae. The subclass, another three is the Stimoniomycetidae. This order, Stimoniomycetidae order, only one order is there. That order name is Stimonitales. This is the division 1. That division name is called the Gymnomycota. In Gymnomycota, two subdivisions are there. In the subdivision, first subdivision, only one class. In that class, two subclasses are present. The second subdivision is two classes are there and three subclasses to present. The second division is called the Mastigomycotina. It includes fungi with observed nutrition, unicellular or filamentous mycelium, cenocytic. You know what is the filamentous body? What is a cenocytic or mycelium? It compresses to, in the Mastigomycota, you can observe to the two types of classes are there, in the subdivisions are there. First subdivision, Haplomastigomycotina. Second subdivision, Diplomastigomycotina. The first subdivision name is called Haplomastigomycotina, includes fungi with uni or biflagellate juice spores are there. In this subdivision, the spores, you can observe that one, single uniflagella or some spores found into the biflagellate juice spores. Next one, this subdivision, three classes are there. The first class, Critidiomycetes. The second class, Hypocritidiomycetes. The third class, Plasmodiaphyomycetes. These three classes most important, that one. First one, the first class is the first class is that one name is called Chytridomycetes. Chytridomycetes, in this one, fungi producing juice spores. Fungi producing juice spores furnished with single whiplash. Furnished with the whiplashed single whiplashed single whiplashed flagella is present instead of at the posterior end instead of the posterior end. The second class, Hypocriteriomycetes. Here the spores motile cells with a single tensile flagella is present interested at the anterior end. This flagella to found into the anterior end. The third class, Plasmodio for Plasmodiophoromycetes. These parasitic fungi. In this class, you can observe the mostly parasitic fungi is producing biflagellate motile cells with both flagella of whiplash type of flagella instead that the flagella to observe the anterior end. Okay. The second subdivision, Diplomastygomycotema. What is the name? Diplomastygomycotema. Sexual reproduction here. In this class, you can observe the sexual reproduction. Oogamous, juice spores, oogamous type of sexual reproductions and juice spores, biflagellate type of juice spores are there. Here, you can observe only one class. That class name is called Oomycetes. In this one, four orders are there. The first order, Laginidales, Saprolaginidales, Leptomytiales, Pernosporales. These four classes are there. First one, Laginidales. Second one, Saprolaginidales. Third one, Leptomytiales. Fourth one, Pernosporales. These four class to observe in Mastigomycotina. Another division 3, A-Mastigomycotina. Here, A-Mastigomycotina, total 4 subclasses are present. Total 4 subclasses is present. That's one fungi absorption nutrition, motile cells is lacking, mycelium type septa or septa. In this division characteristic of fungi, absorption, fungi with absorption nutrition, motile cells, all motile cells is the lacking. Mycelium type is the aseptate type or septate. Here, this includes 4 subdivisions are there. The first is your subdivision, Zygomycotina. Second subdivision, Ascomycotina. Third is the Basidiomycotina. Fourth one is called Deuteromycotina. The first subdivision is the Zygomycotina. These Zygomycotina, you can observe to the, this can observe to the 2 classes you can observe. In the Zygomycotina, how many classes are there? Only 2 classes. One is the Zygomycetes, Trichomycetes. One is the Zygomycetes. The Zygomycetes, it includes total how many? 6 orders are there. In the Zygomycetes, total found into the 6 orders. The second one is called the second one, these Zygomycotina, usually in this one Zygomycotina also, usually Mycelium aseptate, asexual spores non-motile, formed inside a sporangium. Perfect state spores are present. State spores is the juice spores. Here 2 classes are there. One is the Zygomycetes. The second one is the Trichomycetes. Zygomycetes. In this one, Zygomycetes usually saprophytic. Zygomycetes here usually saprophytic, parasitic, or predaceous type. Mycelium impressed in host tissue. This is a characteristic features of the class 1 zygomycetes. This is a very mycelium to impressed in the host tissue. That is a impressed structure of the mycelium present in the zygomycetes class. Okay, coming to the second class, trichomycetes. Trichomycetes, it comprises 5 orders. Here, okay, zygomycetes comprises 6 orders and trichomycetes comprises 5 orders. Here, characters often parasitic or arthropods. This trichomycetes is a parasitism or arthropods. Mycelium not impressed in the host tissue. The two difference here. The first one is a mycelium to attractive to the host cell, zygomycetes, but trichomycetes mycelium is not to attract to the host cell. This is the class, subclass 1. Coming to the second class, Ascomycotina. Ascomycotina here, very important, yeast or septamycelium. Asexual spores, non-motile, not formed inside sporangium. Perfect here inside the sporangium and perfect state spores, ascospores. Perfect state spores meaning what type of spores are they? Ascospores formed as in ascus. Usually within a fruiting body, a ascospores. This class is divided into 6 class. The first class, hemio as hemiascomycetes. Second one is plectomycetes. Third one, hymenoascomycetes. Fourth one, labulobinomycetes. Fifth one is called pyrinomycetes. Sixth one, discomycetes. These all 5 classes. These are all 4 classes. You can observe 6 classes are there, but in here only 5 are there. But another one, 6th class also found into the Ascomycotina. What here you can observe all yeast and septamycelium are present in this class. The first class you can observe, the first class is the that one is the Ascomycetes, the first subclass, not the division is the Ascomycotina division. In the Ascomycotina, subdivisions only 4 subdivisions are there. And see, one is Ascomycotina, Basidio, and Vitido. In the Ascomycotina, total not 6 subclasses are there. The first subclass, hemiascomycetes. Hemiascomycetes or Hemiascomycetidae. Both names are same. Okay. Hemiascomycetidae is no ascospores. In this subclass, compresses 3 orders are there. But here, no ascospores and ascogenous hyphae. SI, naked, thallus yeast-like or mycelium. The thallus structure is the yeast-like or mycelium. This is the first subclass. The second subclass is called Plectomycetidae or Plectomycetes. Plectomycetae, ascospore and ascogenous hyphae, present thallus mycelium asci, uninucleate. Uninucleate and one wall is present. Binucleate, two-walled structure. Fruit body and ascostroma. Fruiting body is called here ascostroma. That is a mass of tissue with lacules. That is a mass of tissue with lacules. The third one, the third subclass, Heminoascomycetes or Heminoascomycetidae. Heminoascomycetidae. This one, Heminoascomycetidae. Here, here 10 orders are there. In this one also, no ascospores, ascogenous hyphae, naked, and thallus, just mycelial like thallus are present. Fourth one, Labulobineomycetes or Labulobinomycetidae. Here two orders are there in this subclass. In this one, two class orders are there. Ascocarp and Ascogenous hyphae present. Thallus reduced. Asci regularly arranged within the ascocarp. This is the perithecium. Unicate in operculate exoparasites of the arthropods is the Labuloneomycetes. Next one is a Lolo ascomycetidae, or this one is called the Laculo ascomycetes. Adi Lolo kadma Laculo ascomycetes. Here ascocarp and ascogenous hyphae present thallus myceliae. Asci binucleate two-walled are present. Fruiting body and ascostroma. That is called the mass of tissue with the lacule. That is the fifth one. And sixth one is the Pyrinomycetes. Pyrinomycetes, ascocarp, ascogenous hyphae, present thallus reduced. Asci unicate, regularly arranged within the ascocarp. Within the ascocarp meaning perithecium. Asci in operculum with apical pore or slit is present. Last in the subclass, sixth one, Discomycetes. Discomycetes is called the, in this one, ascocarp and ascogenous hyphae present thallus mycelium. Asci mycelial, asci unicate, regular arranged in a cup-shaped ascocarp. Asci operculum with the apical spores are present in this subdivision. The another subdivision 3 is the Basidiomycotina or Basidiomycota. Yeast or septamycelium, asexual spores absent in this one. Very, very, very important in this one. The asexual spores are the absent in this fungi. In this class. And this one here present, if present in case asexual reproduction is there, this one is the non-motile perfect state spore, basidiospores formed on the basidium. Okay, here this one is the class. In this one, 3 subclasses are there. One is the Holobasidiomycetes, Phragmobasidiomycetes, Teleomycetes. These Teleomycetes, main order, in this one Teleomycetes or Basidiomycetes, both are the important for this class. One is the Teleomycetes, first one is the Holobasidiomycetes. This basidiocarp present basidia typical arranged in the organized layer. Hymenium, which is completely or partially exposed at maturity. Basidia septae or aseptae. Basidiospores all ballistospores. That is all the blastospores. Next one is the Phragmobasidiomycetes. Phragmobasidiomycetes, basidio present basidia typically arranged in organized layer hymenium, which is the completely or partially exposed at maturity of the Basidia septa or aseptic condition. These basidiospores is the ballistospores. This one is called the ballistospores. The last one is the teleomycetes. Basidiocarp lacking teleospores. This is a encrusted. This one is called the encrusted proboscidea, grouped in a sori or scattered within the host tissue, parasitic or vascular plants. This one is the three. Last subdivision is called Deuteromycotina. Yeast, in this one, Deuteromycotina, here yeast, septum mycelium, asexual spores as in the Asphomycotina, perfect state. Spores also here absent, very rare and unknown. In this one includes imperfecti fungi. This one is the perfecti fungi, sexual stage unknown. It compresses in the one class, that is the Deuteromycota, Deuteromycetes class. In this one, three subdivisions are there. C subclasses. One is the blastomycetes or blastomytidae. Second one, hypomycetidae or hypomycetidae. Third one, poliomycetes. The first one, the first class, blastomycetes. Blastomycetes is the budding. Yeast are yeast-like cells with or without pseudomycelium, true mycelium lacking or not well-developed mycelium in blastomycetes. The second one, the coleomycetes. Mycelial asexual spores formed in a flask-shaped structure that is called pycnidium. Flask-shaped structure is called the pycnidium or on a pod. Fungal tissue, acervulus. Acervulus. Fungal tissue is called the acervulus fruiting body is present in this one. The third one is called hypomycetidae. Mycelial, sterile or bearing asexual spores directly on hyphae or on special branches conidiophorus. This one is the total three subclasses. In this one, some 63 lakhs 5,000 species of fungi have been described with another 13,500 associated with algae as lichens. The Martin scientist in 1965 proposed a classification according to the spore formation in the fungi. This is the total classification. See, division, mycota. The two subdivisions, the Martin scientist dividing the like this. The two subdivisions also present in this one. The subdivision one, Myxomycota. The second subdivision, Eumycota. Myxomycotina under the all bacteria like this only. But this Eumycotina, again two division. One is a lower plants and second one is the higher plants. That is a lower fungi, higher fungi. Division mycota. This includes non-green, nucleated, talophyte, which are saprophytes or parasitism in nutrition. Okay. Before the class explanation, what is the characteristic feature of the mycota? Mm, mycota and this one mycota, non-green nucleated thallophyta which are saprophytic parasitism in the nutrition. Subdivision 2, one is the Myxomycotina, second one is the Eumycotina. Myxomycotina, here this Myxomycotina, thallus is achlorophyll. Myxomycotina characteristic. Thallus is achlorophyllous, multinucleated mass of protoplasm called plasmodium. These are the Myxomycetium is the plasmodium. The example slime molds are present. Coming to the subdivision second, Eumycota. Here, all the fungi except the smile mold are included in this subdivision. This is the lower fungi. This subdivision can be divided into two parts. One is the lower fungi. The second one is the higher fungi. Lower fungi, they have simple thallus which are unicellular and other filamentous. Mycelium usually septa is not present. What is the lower fungi? They have lower fungi, they have the simple thallus. Lower, very simple thallus is there. All are the unicellular and other filamentous is the mycelium. Usually septae is not present in the lower one. In this lower, motile cells are produced. Here lower fungi can be divided two parts. One is the motile cells, second one motile cell lacking, non-motile cell. In the lower fungi, some fungi is the motile, some fungi is the non-motile or lacking cells. One motile cells, this one lower fungi, unicellular, other filament are there. First one is the motile cells. Here motile cells again to the four classes are there. Critidiomycetes, first one. Second one, hypocritidiomycetes. Third one, plasmodiophoramycetes. Fourth one, oomycetes. In the motile fungi, two classes. One is the zygomycetes, second one trichomycetes. What is the class critidiomycetes? This is the motile cells have single flagella, whiplash type of flagella inserted at the posterior end. The second one hypocritidiomycetes. Motile cells possess a single flagella of tensile type of flagella instead at the anterior end. Third one, plasmodiophoramycetes. Motile cells are biflagellate, that is a whiplash type, but one is long, another one is short, that is a whiplash type. Last class, oomycetes. Here, motile cells are biflagellated cells, nearly equal length, one of the point forward and the other trails behind. These are the motile cell in the lower fungi. Another one, non-motile or motile cell lacking zygomycetes. Here, motile cell totally absent. Asexual reproduction takes place by sporangiospores. This is the Zygomycotina class explanation. The second class, trichomycetes. Here also motile cells are lacking. Asexual reproduction takes place by the conidias. This is the lower fungi can be divided into motile and non-motile. Motile again four subclasses, non-motile two classes. And coming to the higher fungi, in stomatic phase, consists mostly of a mycelium which is usually septate. This one divided in three classes. One Ascomycotina, Basidiomycotina, Deuteromycotina. Ascomycotina characteristics spores are called ascospore, are produced endogenous with sac-like structures are called asci. The second one, Basidiomycetes. Characteristic spores are called basidiospores, are produced exogenous on club-shaped structure called basidia. The third one, Deuteromycotina or Deuteromycetes. Sexual stage is unknown. Here, sexual stage is totally absent, Deuteromycotina. And this is unknown. The stomatic phase consists a septa mycelium which multiply by the conidia. This is the higher fungi classification. a more natural system classification. This is the which scientist to explain? Martin scientist in 1965 explanation of the type of classification. Again, the another classification, Ainsworth classification. Very, very important. Ainsworth, this is the expected question for the fungi. Ainsworth classification, a more natural system of classification of fungi. This one more classification of fungi was proposed by the Ainsworth in 1973, which has been accepted by many mycologists, many mycologists today, like Webster, another scientist, many scientists to accept the this type of accepted with this type of mycologist today. Webster 1980, Bill Grammy 1985, and Dubé 1987 to accepted to the this type of fungi classification. See here, what is the kingdom? First one is the mycota is the kingdom. Division Myxomycotina, Eumycotina, both same. In the last scientist to explain like this. Myxomycotina and the Eumycota. Myxomycotina, again two classes are there. One is the Myxomycetes. Second class is the uh, acroisomycetes, and another class is the heteroxylomycetes, another plasmodiophoromycetes. Myxomycotina, total four classes are there. Myxomycetes, Hyrelio, exomycetes, Plasmodiophoromycetes, Acrysomycetes. In the division of the Eumycota, see, division of Eumycota, total four subdivisions are there. The first subdivision, Mastigomycotina, Ascomycotina, Basidiomycotina, Zygomycotina, Deuteromycotina. Mastigo, Asco, Decidio, Jaigo, and Deuteromycotina. Mastigobicotina divide into in this subdivision. Divisions of the three classes are there. One is the Criteriomycetes, Hypocriteriomycetes. Third class is the Oomycetes. You can by heart this total slide, very, very important. The second subdivision, Ascomycotina. Ascomycotina, the first class, Enescomycetes, Laculoascomycetes, Labillobleneromycetes, Platinomycetes, Pyrinomycetes, Discomycetes. Discomycetes. This is the Ascomycotina subdivision. Next, third subdivision, Basidiomycotina. Class Teleomycetes, class Hymenomycetes, Gastromycetes. In Basidiomycotina, three subclasses are there. Zygomycotina, only two subclasses. One is a Zygomycetes, second one is a Trichomycetes. Next one, Deuteromycotina. Three classes are subclasses are there. One is the Blastomycetes, Ciliomycetes, Hypomycetes. This is the total Ainsworth scientist to explain the classification. Classification based on the spore production, on the basis of the organism, the vegetative thallus morphology, and the reproductive structure, way of the spores produced, and particular life cycle of involvement, kingdom of mycota is classified into following division. Phycomycotina, Oomycotina. Phycomycotina, it includes the simplest fungi type of fungi. It is also called algae fungi because of the characteristic of them are similar algae like Vachidia. See, that's why this is a phycomycetes. In between this phycomycetes, in between the both algae and fungi. both algae and fungi. This because most of the characteristic of them are similar to algae like Vachidia. They have simple thallus, which is unicellular phenotype, xenocytic, accepted or accepted filamentous. The reproduction reproduced asexually by the formation of zoospores or non-motile spores. Sexual reproduction is isogamous or heterogamous, which take place by gametophysial contact. The diploid phase is represented by the zygote. Phycomycetes has been classified into the two subclasses. One is the oomycetes. The second one is the zygomycetes. The characteristic features of the oomycetes. It range from the primitive unicellular thallus to a profusely branched filamentous mycelium. Many members of them are terrestrial and obligate parasites. Asexual, they reproduced by the biflagellate zoospores. Biflagellate zoospores are present. Oogamous type of reproduction. The sexual reproduction is a oogamous reproduction that involves the fusion of the male and female gamete to form to the oospore. Oospore undergoes after meiosis division to produce the haploid biflagellate zoospores. Example, Phytophthora infestiana. This is causing by the disease. This fungi, leaf blight diseases. Phytophthora infestians causing the disease in potato plants, leaf blight of the potato. Coming to the second one, zygomycetes. The group is named zygomycetes because the diploid resting spores are called zygospores. That's why zygospore is formed during the life cycle. They are mostly saprophytic. They are mostly saprophytic. Some other are parasitic on plant and animals. Plant and animals. The vegetative body is mycelium. Body is mycelium, which is well developed, profusely branched and phenotype. The absence of the absence of motile sexual or asexual cells are present. And next one is the sexual reproduction takes place by sporangiospores, aplanospores, or by sporangiospores, aplanospores, or by the conidia. These are the asexual reproduced. And the next one is the sexual reproduction occurs by conjugation of gametangia, resulting in the formation of the spores. The best example spore is called the rhizospore or nucar is the best example for the zygomycetes. Next one, ascomycetes. Coming to the ascomycetes. The species of ascomycetes are called the sac fungi. This is called the sac fungi because they produced sexual reproduction of the spores within the sexual reproduction of the spores within the sac-like variations. Sac-like vascus are present. Ascomycetes are mostly terrestrial fungi, mostly terrestrial and occurring as sporophytes or parasite. They have well-developed branched septa, septae mycelium, except yeast. Yeast is the unicellular fungus. Asexually, they reproduced by the non-motile spores, Conidia, Odia, or Chlamydospores. The spores name is called Conidias, Odia, or chlamydospores. Sexual reproduction takes place by fusion of the gametangia of opposite mating type. Here, there is absence of motile cells. In Ascomycotina, the motile cells are absent. Example, Saccharomyces cerevisiae, yeast. Saccharomyces is the yeast. Saccharomyces cerevisiae, Penicillium, Aspergillus, under in the Ascomycotina. These are the four very economically important Aspergillus to causing the disease from the Solanaceae family, tomato, chili, like this. Penicillium, you know, Penicillium notatum. This is a antibiotic. Okay, to prepare the Alexander Fleming to that one. Yeast, you know, yeast is usually you preferred for the many industrial microbiology used for this yeast, different uses for the yeast. Coming to the Basidiomycetes, the members of Basidiomycetes are saprophytic or parasitic. The group is named Basidiomycetes as they produced the basidiospores, the club-shaped basidium during the asexual reproduction type. Club-shaped during the sexual reproduction. Mycelium is highly developed profusely, main branched and septae is present. The mycelium are differentiated in two mating types. In the two types are there. One is the positive mycelium, another one is the negative mycelium. There are two kinds of mycelium, primary mycelium and secondary mycelium. Primary and secondary mycelium. Asexual reproduction takes place by fragmentation method, budding method, Odia method, Conidia method, or chlamydospore method. Okay, the dikaryotic cells is formed during sexual reproduction. Sexual reproduction here, absence of motile cell throughout the life cycle. These are the most advanced fungi as their fructifications are often large and prominent. Very, very interesting Basidiomycotina. Economically, example for economically use for the mushrooms, edible mushrooms. Uh, Very important, mushrooms, edible, high protein is present in the mushroom. Another one, two are causing the disease. One is a Fuchsinia, second one Eustilago. Fuchsinia is causing tea leaves, or Fuchsinia and wheat leaves also. Tea leaves and wheat leaves. Eustilago also causing the different diseases. This is a Basidiomycotina. Coming to the Deuteromycotina. Deuteromycotina comprises more than 1700 species of the diverse habitat. It is considered as an artificial class of fungi. The class of Deuteromycotina are sporophytes as well as parasite. Both are there. Sporophytes are as well as parasites. Parasitic fungi causing serious disease to plants, animals, include human being also. That's why this is the imperfecti fungi. This is the important character from the Deuteromycotina. Deuteromycotina, imperfecti fungi. Mm. In Imperfecti fungi is the which fungi is the imperfecti? That is the Deuteromycotina is the imperfecti fungi. Some of them uninuclear, while other of the multicellular structure. They reproduced asexually by conidia, along with some other two types of spores are present. The sexual reproduction is earlierly absent in Deuterobricotina. Very, very important. The sexual reproduction is absent. The asexual stage or imperfecti stage, imperfect stage in Deuteromycetes is well defined, but the sexual or perfect stage is absent in life cycle. Therefore, they are called fungi imperfecti. Very, very important. What is the fungi imperfecti? The sexual stage or imperfecti stage is Deuteromycosis is well defined. Is well defined, but the sexual or perfect stage is absent in life cycle. That's why there is called this fungi, Deuteromycotina fungi is called imperfecti fungi. What are the example? Alternaria, Fusarium, Helminthosporium. This disease causing human beings, animals, plants also. Human beings is the Alternaria causing in tomato plant. Many disease causing the disease Alternaria in the mostly Solanaceae family, that too tomato. Okay. Fusarium, you know, many causing the disease in the human beings, Fusarium, Helminthosporium, both are. This is the Deuteromycotina. Coming to the classification based on medicinally important of the our fungi. Okay, what is the okay, in the based on the classification, what is the importance of the fungi? Mycos are classified superficial, cutaneous, subcutaneous or systematic infections depending on the type of degree of tissues involved and the host response the pathogen. The first one, superficial mycosis intent. Superficial mycosis intent occur in the tropics and restricted to outer surface, hair and skin. This is the first one. Example, Pediaria hydrate. This is the tinnitus. Second one, Cutaceous mycos. These are the three genera of fungi. Three genera of the fungi. The first one, Microsporium. Excuse me. Second one, Trichophyton. And third one, Epidermophyton. That commonly causing disease in the non-living tissues of skin, hair, nails, claws, people and animal by growing in a zone just above where the protein creatine is deposit. In that place, you can observe this type of fungi. Next one, subcutaneous mycosis. They are normally saprophytic. They are normally saprophytic inhibition of the soil, particularly in the typical and subtropical area of the Africa, India, and South America. This is the third one. Fourth one is the systematic mycosis, or in infection that the whole body of the plants, animals, or human being. We divided these into mycosis due to primary usual the dimorphic virulent pathogens are there, and those due to opportunities of the pathogens. These are the classification based on the medicinally important of the fungi. Next classification based on the rotate of acquisitum. This one is the infecting fungi. Infecting fungi may be either exogenous or endogenous. Both infections are there. Exogenous or endogenous. When classified according to the root of the acquisitions, a fungal infection may be designated as exogenous or endogenous of in origin. If classified as exogenous, an infecting organism may be transmitted by airborne, cutaneous, or percutaneous rate. This is the exogenous. In case endogenous acquired fungal infection may be acquired from colonization, reactivation of fungus from talent infections are there. Okay, these are the next classification based on the virulent. Classification based on the virulent. Primary pathogen can establish the infection in normal host. In normal host. Uh, Next, opportunities pathogens causing diseases in individuals with comprised host defense mechanisms. This one, deep mycosis are causing primary pathogenic and opportunities fungal pathogen. The primary pathogenic fungi are able to establish the infection in normal host. In normal host. Whatever opportunities of pathogenic require compressed host in order to establish the infection. That is the cancer, organ transplantation, surgeries, and AIDS. This is the infection normal pathogenic fungi able to establish infection the normal, whereas the opportunities fungi require the host in established function, where this is the primary deep uh, deep pathogen usually gain access to the host via the respiratory tract. Opportunistic fungi causing the deep myces invade respiratory tract, alimentary tract, or intervascular device. Okay. Next one, the primary systematic fungal pathogen include coccidials, immunity. Next one, histoplasma capsulatum. Next one, blastomycetes determites and paracoccidials bacilli. These are the example. The opportunistic fungal pathogen include Cryptococcus neoformasis. Next one, Conidia aspergillus species. Pencilia marinife. Example this. The Zygomycetes, Trichospora, Bacilli, and Fusarium species. These are the this one. What is the key point? This total key point of this one. Here, fungi is a plural word. Fungus was the derived from the Latin word fungaro. And fungi are achlorophyllus, heterotrichous, achlorophyllus, heterotrichous eukaryotic thallophyte. Next one, branches of botany that deals with fungi is called mycology. And the scientist who is cornered with the fungi is called concern with the fungi is called mycologist. P.A. Mitchell, known as after their mycology, Butler, father of Indian mycologist. Fungi are eukaryotic organism, means they have true nucleus which are enclosed in membrane. They are non-vascular organism. By here, xylem and phloem both are absent. Fungi have cell. Plant also have cell wall, but animals have no cell walls. There is no embryonic stage of for fungi. The reproductive by means spores. They are sexual, asexual spore. Sexual spores is the are oospore, zygospore, ascospore, basidiospore, etc. Asexual spore, sporangia, sporangiospores, aplanospores, zoospores, conidia. Depending on the species and conidiation, both sexual and asexual spores may be produced. They are typically non-motile. Fungi exhibit the phenomena of alternation of generation. They have both haploid and diploid stage. The another one, the fungi are acrophilous. Acrophilous meaning chlorophyll is totally absent. That is the acrophilous, which means they lack of chlorophyll pigment present in the chloroplast in plant cell, and which are necessarily for photosynthesis. Uh, as they are achlorophyllous, therefore, they do not have the ability to make their own food material. That is a heterotrichous. These are the not eutrichous, heterotrichous plant body. The vegetative body of fungi may be unicellular are composed microscopic thread-like hyphae. Hyphae can grow and form a network called mycelium. Yeast are unicellular fungi that do not produce hyphae. The structural of the cell wall similar plant but chemically fungi cell wall composed of the kinetin. Kinetin formula C18H38 13O5N. This is the fungal cell wall to contain chitin. Fungi are heterotrophic organism. You know that one is called the depending other organism, food material depend the other organisms. They obtained its food and energy from organic substance, plant and animals matter. Fungi digest the food first and then ingest it to accomplish this is the fungi produced exoenzyme like the exoenzymes like hydrolase, ligase, oxidoreductase, transferase, etc. These fungi, their food is called starch. Fungi are saprophytes. Saprophytes meaning get energy from the dead and decaying matters. This is the saprophytic fungi. Very important. Fungi different. Saprophytic, they get energy from the dead body and decaying matters. Parasiting, live in a host, attack and kill. This is a parasitic. Symbiosis, mutually beneficial symbiosis. Next one, optimum temperature of growth of the most saprophytic fungi
20 to 30 degrees centigrade, while the 30 to 37 degrees centigrade also for parasitic fungi. Growth rate of fungi is slower than the of the bacteria. The very slow compared to the bacteria. Fungi growth rate is less to compare to the bacteria. Reproduction in fungi is both by the sexual and asexual means sexual state is referred to as telomorph, that is a fruiting body. Asexual state is referred as the anomorph, mold like. And asexual reproduction methods are fragmentation, stomatic. The first one is a fragmentation, stomatic budding fission, asexual spores formation. Sexual methods are gamete copulation, gametangium, gametangium copulation, stomatic copulation, and spermatozation. Some fungi are macroscopic and can be seen by naked eye. Mold or mushrooms are the best example macroscopic fungi. Okay, in 1991, landmark paper esteemed that are 1.5 million fungi on the earth. See here, how many fungi are there? 1.5 million fungi. Only about 300 species of fungi are infection human. 1.5 species affected from human, 300 species, only about 300. The classification of fungi designed mainly of parasitical application, but it also bears some relation phylogenic consideration. The fungi can be classified according to the various parameters. Various parameters include the classification based on taxonomy, hierarchy, classification based on spore production, classification based on medicinal important, classification based on acquisition, classification based on virulence. This is the basic thing, okay? Alexophora Mimus proposed fungal classification 1979. They placed the fungi include the slime mold in the kingdom Mycete. They divided kingdom Mycete into three divisions: Gymnomycota, Mastigomycotina, Mastigomycotina. Martin scientist in 1965 proposed a classification according to the above spore formation of fungi, divide and division of mycota into two subdivisions, mycotina and eumycotina. Based on the classification, medicinal important fungi divide the fungi into superficial mycosis, subsane mycosis is the systematic. This is the total key points for the fungi. In the fungi, some terminology also most important. AM. AM meaning Arbusculus mycorrhiza, A mycorrhiza or AM mycorrhiza, where fungi from the Glamoromycota penetrate the root of plant and provide the plant with a water and nutrient the sugars of the fungi. Ascospore. Ascospore is the fruiting body of the Ascomycotina. Ascomycete is a class of fungi that produced their spores in the sac-like cells is called asci. Next one, ascospores. Sexual spores produced in the asci of Ascomycetes fungi. Ascus, the spores producing cells of an Ascomycetes fruiting body. Basidiocarp. Basidiocarp fruit body is a Basidiomycetes fungi. Basidiomycetes also a class of fungi produce their spores to Basidia. Basidioclarp is the one type of fruiting body of a Basidiomycetes fungi. Next one, Basidiospores is the sexual spores produced in the Basidia. This is the Basidiomycetes fungi. Basidium, this is the spore producing cell of Basidiomycetes fungi. Next, cellulose. Cellulose is a component of plant cell wall and of wood composed of glucose unit. Next, chlamydospores. Asexual spores formed by the breaking of fungal hyphae, clamp connection, swollen area formed around septa in a hyphae during cell division. Next, corporophylla growing on dung. Cuticle, the surface layer of cap or stem of the fruiting body. Next, Deuteromycetes. Deuteromycetes, absolute term of group fungi not reproduced asexually. Asexual reproduction, sorry, sexual reproduction. Sexual reproduction total absent in the Deuteromycotina. This Deuteromycotina dichotomous, this dichotomous forking ascomycety for forking and divided into pairs is an logical decisions making trees. Dictobus, forking divide into part as a logical decision-making trees. Dichorion, a pair of closely associated sexual complete nuclei. Next, ectomycorrhiza. Ectomycorrhiza, where the fungal form sheathal around the rootlets, growing between, not penetrate in the cells. That is a absorption of water nutrient. Next, endomycorrhiza, endophytic, hyphae, inferior, lichens, mucilaginous. These are the expected questions. These are the expected questions, and this one, thank you. The total fungi are the complete in the session.